This is a production of Cornell University.
All right, thank you, Larry. Yeah, and thank you all for attending my exit seminar. So here's a quick overview of what I'm going to cover in this presentation. So we'll start off with a quick background on sex determination and the Salicaceae family, which is what includes both the willows and the poplars. Then we'll move into how we try to understand the genetic mechanisms of sex determination in shrub willow. So we started with this sort of integrated genomics and bioinformatics approach to identify candidate sex determination genes. We then refined some of those hypotheses using monoecious willows and families and sequences we generated from those. And then we functionally validated our candidate genes through overexpression in Arabidopsis and DAPC. And lastly, I'll finish off by talking about a couple of other projects that I've worked on, one involving the de novo assembly and annotation of 11 shrub willow genomes, and another on how we developed a protoplast transient expression and knockout assay to enable future functional genomic studies in shrub willow. So a quick background on willow. So willows are the genus Salix. These have over 300 species. These include the tree willows, which you might be most familiar with, that you see around in parks and gardens. Also the shrub willows and the Arctic dwarf willows. Now in the Smart Lab, we are primarily interested in the breeding and genetics of shrub willows for biomass and bioenergy production. So in a bioenergy production, they're grown in high density plantings shown here, and they're composted every 2 to 4 years. And these wood chips are then burned to generate heat and electricity. However, shrub willows also have several other applications. So they're used as ornamentals, also in ecological restoration, and then in some traditional uses like basket. So one key feature of the willows are that they are dioecious. They have separate male and female plants, and that's primarily where my research comes in. So here are the female catkins here on the left, and these are the male catkins on the right. So catkin just refers to this whole inflorescence here. And my research specifically focuses on the species Salix purpurea as a model for the shrub willows, and which had a chromosome 15 ZW sex determination system. So all that means is that chromosome 15 is the sex chromosome, and the females are ZW and the males are ZZ. So the W is only inherited to the female line. Notably, willows are also the sister genus of the poplars or populus. So populus trichocarpus is the model tree. So we could leverage some of the genomic resources available from that in our study of willows as well. Now, getting into sex determination and sex chromosomes, which is where my research focuses. Sex chromosomes are characterized by these large non-recombining regions. So, and these are called sex determination regions, or SDR. So I'll say SDR in this talk, and that's what I'm referring to, is that large region that we don't see recombination. And one of the challenges is these sex determination regions can contain hundreds of genes. In the case of Salix purpurea, there are 488 annotated genes on the chromosome 15W SDR. Now, adjacent to the SDR are pseudo-autosomal regions, which are freely recombining regions of the sex chromosomes that can be treated like autosomes for all intents and purposes. And then when we look at inheritance of sex, there's two primary systems in angiosperms. There's the XY and the ZW. And in the XY system, the males are the heterogametic sex, so males are XY, and females are the homogametic sex, so females are XX. This is the system that's present in humans and mammals, so you're probably familiar with that. ZW, on the other hand, just means that males are ZZ and females are ZW. So the W is only inherited to the female line. And this is the system that's present in the shrub willows. So then when we think about how sex is controlled as far as genes and mechanistically, there are two leading models, the single gene and the two gene model. And this basically means that only one or two genes are both necessary and sufficient to switch sex from one to the other. So there's I have pictured here a couple of examples of these single gene and two gene systems, right? So a good example of a two gene system is asparagus, where females are XX, and this results in female floral development. On males, which are XY, on the Y chromosome, they have this tapetum development function 1 gene that promotes male floral development. And linked to that, they have a suppressor of female function gene that suppresses female floral development. So you get male flowers. An example of a single gene system is persimmon. And that's actually kind of a really interesting system. So females are XX, and then there's this autosomal gene called MEGI. And MEGI promotes gynoecium development, which results in female flowers. MEGI also promotes SVP expression, which suppresses pistillata. And because it suppresses pistillata, you don't get male floral development, so you get female flowers. Males have this Y chromosome, which has an OGI, which is actually a small RNA. And this small RNA is the only gene that's necessary to switch sexes. So it does that by methylating the promoter of MEGI, therefore turning off the expression of MEGI. And as a result, you don't get expression of gynoecium, but you do get expression of pistillotic, and you get androecium and male floral development. So when we're thinking about how do we understand sex determination genes in shrub willow, we have this challenge of almost 500 genes in the sex determination region that are fully linked, so we don't have recombination. And that's as plant breeders, we really leverage recombination. So how do we parse out those almost 500 genes to find the one or two that are responsible for sex determination? And that's really what my dissertation has focused on. So now we get at sex determination and Salicaceae family. So the Salicaceae family includes the poplars or populus and willows, salix. And I have this phylogenetic tree here. This was generated by one of our collaborators at Texas Tech, just to show you some of the relationship between some of the populus species and some of the salix species. And section Vetrix here highlighted in red, these are the shrub willows that we use in our breeding program. And that's where Salix burkhardia is. So what's interesting is that this entire family is dioecious. However, the sex determination region has actually shifted both heterogametic systems and chromosomes in this family. So in poplars, there's chromosome 19XY, chromosome 19ZW, and chromosome 14XY systems. In willows, on the other hand, so in the tree willows, chromosome 7XY is the system. In some of the basal shrub willows, it's actually a chromosome 15XY. And then in the shrub willows or Vetrix and the dwarf willows, Chamaita, all these, it's chromosome 15ZW. So we can see this system has changed multiple times. So notably in poplar, AR17 has been implicated as the single gene master regulator of sex throughout this genus, such that its presence and expression results in female, and a lack of expression either through its absence or through its silencing results in male floral development. So and this appears to be conserved across populus. However, we're really interested in trying to see, is this gene also involved in willow? Is it not? How is the system different in willow? Because clearly this sex determination system has changed multiple times. And so we really want to characterize that further in the willows and specifically the shrub willows. So to start off, our first step was to try to get a big picture of what does sex dimorphism look like in shrub willows. So what we did, we took this, what we're calling an integrative genomics approach. We took 90 male and 90 female catkins in F2 family. We did GDS resequencing on every individual. We also did RNA-seq on each one of those catkins, and then small RNA-seq and bisulfite sequencing to look at CpG methylation on a subset of those as well. And so we obtained all this data, and that's actually summarized in this circles plot here. And there's a lot going on here, and you definitely can't see all of it, but I just want to give you a big picture of sort of the depth of data we collected. And one could really spend a better part of a career parsing through all the data from this. But of course, we're really interested in sex determination. So because we collected both genotype data and expression data, we were able to do an eQTL analysis, which is where you associate expression of genes with polymorphisms elsewhere in the genome. So in this circle plot here, this outer track, these are the relative numbers of eQTLs per locus from our study. And then in this inner circle here, in turquoise, I have the genes that have eQTL mapping back to the 15W STR, and in gray, the genes who have eQTL mapping back to the 15Z STR. So we can see there are thousands of genes whose expression is associated with genotypic polymorphisms in the sex determination region. This middle track here shows the total differentially expressed genes. So we detected over 24,000 differentially expressed genes between male and female catkins. This is not entirely surprising given the morphological differences that we observed between catkins. And then this inner track here, these are the differentially methylated regions. So we detected, this is CpG methylation, which we detected using bisulfite sequencing. So once again, blue for male, magenta for female, and then those in yellow here are actually differentially methylated regions in the putative promoter regions of genes. So one really interesting finding from this was we saw about threefold higher methylation in male catkins relative to female catkins, which was very interesting. And then we also did small RNA sequencing. We detected 45 microRNAs. Of these, 5 actually are unique. They haven't been identified in any other species. And then I have their top 10 target sites in purple here. So a lot of data from this. Another key finding we found was that among these eQTLs, so these genes that had associations back to the SDR, we identified 97 transcription factors. And we were really interested in these because transcription factors control the expression of many other genes. And these could represent sort of intermediate pathway genes between the 1 or 2 sex determination genes and the 24,000 differential expressions by controlling their expression. So we wanted to look at some of those. We also looked at sex dimorphism related genes and their associations through eQTL with the SDR. So primarily floral development genes, which are involved in male or female flower development and thus primary sex dimorphism, and also secondary metabolism genes. So things like terpenoid, flavonoid, and phenolglucoside. These compounds have all been shown to be differentially produced between male and female cactus. So we found that many MASBOX genes, as well as these secondary compound genes, had eQTLs with the SDR. And this was even after we accounted for sex as a covariate in our statistical analysis. This figure here, this shows the flowering pathway genes that have been characterized in Arabidopsis. I have the number of paralog or number of homologs in the willow genome for each of these genes. And then they're purple if there are copies that are male and female differentially expressed, blue if they're male, and red if they're female differentially expressed. So you can see what differential expression looks like in the male and female floral development pathway. And then those highlighted in red here actually have eQTL mapping back to the SDRs. They represent potential regulatory entry points for the sex determination region into the floral development pathway. And then, of course, we used this genomic study to identify candidate genes for sex determination, genes we think might be the master regulator genes for sex. So we looked at genes that were present in the chromosome 15W SDR, so present in females and absent for males, those that had very high levels of expression in females, those that were, we also did network analysis. We identified genes that were in the female correlated network. And then lastly, we looked at their functional annotation to see if they could potentially have a role that would involve sex determination. So we identified this short list of genes here. So you'll see in the short list, we actually have these 4 tandem repeats of AR17, a cytokine response regulator. If you'll recall, that is the master regulator gene in poplar, and it's showing up again from our analysis as being a strong candidate for a sex determination gene in willow. However, we see several other genes that might be interesting as well. So there's this GATA15 family transcription factor. This is involved in floral transition and morphology in Arabidopsis. We have a zinc knuckle transcription factor. We have a truncated Argonaute-4 gene. So Argonaute-4 is involved in the RDDM pathway, the RNA-dependent DNA methylation. So it could be responsible for this threefold difference we see in methylation between males and females. And then also a few hypothetical proteins. So we developed a short list, but we know only 1 or 2 of these are actually going to be involved in sex. So we really need to further narrow down this list. So our next step then was to refine our sex determination hypothesis and validate this using monoecious willows, which represent a natural disruption of a dioecious system. So very rarely, monoecious willows can be found in nature. And we have one such individual in our collection, which we call 94003. So this plant actually has separate male and female flowers on the same catkin. So it's monoecious. And we developed an S1 and an S2 family, as well as a male and female outcross from each. So we crossed this monoecious parent to our male reference genome and also to our female. And so we obtained these 4 families that we used to look at the inheritance of monoecious. We also conducted Illumina and PacBio sequencing of this monoecious parent, which we used to annotate and assemble a monoecious reference genome. So now we have a monoecious reference in addition to our pre-existing male and female reference genomes. We also conducted Illumina DNA and RNA-seq of some of the progeny from this monoecious parent. And from that, we identified the causal variant responsible for monoecious. So notably, monoecious plants are genotypically ZW. So if you'll recall, females are ZW, males are ZZ. So it appears that monoecious willows are genotypically female. However, their chromosome 15W has this large 1.1 megabase deletion on it. And that deletion actually disrupts most of these candidate sex determination genes. So the 15W in monoecious willow is missing AR17 and many of these others. In fact, GATA15 is the only one of these that's still present. So that tells us a couple of things. One, if AR17 were a single gene master regulator that is responsible for switching the sexes like it is in poplar, its absence would mean that these plants would be male. That does not appear to be the case. And the fact that we get these monoecious intermediates suggests that it's likely a two-gene system regulating sex in willow, such that we have a partial disruption, so we get this monoecious phenotype. And so this is what that monoecious parent genome assembly looks like. So here is a dot plot. This compares alignment between two genomes. So where you see a line or a dot, that's where there is a similar sequence. So on the x-axis, we have our female reference genome, chromosome 15W. And then on the y-axis, we have our contigs from our PacBio assembly of our monoecious parent. So in the middle here, this track is actually our chromosome 15W. It was assembled into a single contig, which is quite nice. And then our chromosome 15Z in the monoecious parent was assembled into these two contigs. So you'll see where you don't see mapping from the Z contigs. This is the sex termination region. And then highlighted in yellow here, this is actually that 1.1 megabase deletion. So we see that this region is not present in any of our chromosome 15 contigs in the monoecious parent, but it is in our female reference. So this large deletion is disrupting AR17, but GATA15 is the only one of those candidate genes that's still present. And so that tells us GATA15 might have a role in sex termination. So I mentioned we generated self and outcross families from this monoecious parent. So because this monoecious plant is ZW, but this W has this large deletion, we're choosing to call this W chromosome a W superscript H, such that it results in monoecious plants instead of female. So when we self our ZWH monoecious parent, we would expect 25% ZZ male, 50% ZWH monoecious progeny, and then 25% homozygous WH progeny. Now, when we look at the S1 and the S2 families, we see they're segregating for roughly around 2 to 3 monoecious to 1 male. And we also collected GBS resequencing data on every individual in this S2 family. And we only saw ZZ and ZWH genotypes corresponding with male and monoecious respectively. So that tells us that homozygous WH is actually lethal. So this large 1.1 megabase deletion is probably disrupting genes critical to function in plants such that they don't survive. So to validate our hypothesis, we outcrossed our monoecious parent, ZWH, to a male, ZZ, and we got 50% monoecious and 50% male progeny, as we would expect, validating this hypothesis that we observed. Now, things get a little more interesting when we crossed our ZWH monoecious parent to a ZW female. So we saw about 25% male, 25% monoecious, but then about over half this family was actually female. And among these females, from our Illumina DNA resequencing, we saw both ZW and WWH females. So that tells us that a normal W can actually restore viability that is lost when you have a WH. And so looking at that closer, these are IGV plots of our Illumina DNA resequencing from that monoecious by female cross. So that family that we saw, males, females, and monoecious progeny. And what this is this represents the relative amount of reads mapping per locus to the reference. So at this top, this is our female parent 15W reference, and the bottom here, these are mapped to our monoecious parent 15WH reference. So in the males here highlighted in blue, we see they don't really have much mapping to either the W or the WH chromosome, which makes sense because they are ZZ genotypically. The monoecious plants highlighted here in green, they don't really have any mapping to the female 15W, but they do have pretty good mapping to the monoecious 15WH, confirming their ZWH genotype. And then for the females, we see all 5 have mapping to the regular chromosome 15W from the mother, confirming that chromosome is necessary for female in this cross. But when we look at their mapping to the 15WH monoecious reference, we see 1 actually has pretty good mapping and the other 4 do not. So that tells us that 4 of these are ZW, and this 1 individual here is actually WWH, confirming that genotype is viable. So I started off by showing you this integrated genomics approach, how we use bioinformatics to identify candidate genes, and then how we refine this hypothesis using monoecious willows. But ultimately, what we need to do is functionally validate our genes. Now, unfortunately, in willow, no one has figured out how to successfully regenerate willow from tissue from callus culture. So we can't generate stable transplants. So because of that, we have to rely on a heterologous system to test our candidate genes. So we chose all of our favorite model plants, Arabidopsis, to overexpress each of these candidate sex termination genes. So we overexpress these in Arabidopsis, and then we followed it up with floral proteomic analysis. So in doing floral proteomics, we could look at what genes or pathways are perturbed when we express each of our candidate sex determination genes. So this list here, I show, these are 6 of the candidate sex determination genes that we tested. We also tested leafy and flowering locus T as control genes. So these are genes that we know are not involved in sex determination per se in willow, but they are involved in the floral development pathway, and we know we should be able to get phenotypes from these when we express them. We got around 4 to 6 independent events for each of these overexpressed genes, and so we detected anywhere from 11,000 to 16,000 proteins from each of these overexpression events. And if you'll notice from the total differentially abundant proteins, these are the number of proteins that have significant differences between each overexpression line relative to the empty vector control. We see that 3 of them have a very large number of differentially abundant proteins. So AR17, GATA15, and then this hypothetical protein all have over 4,000 differentially abundant proteins in floral tissue. So that tells us that they probably have some role in floral expression. And when we look at these 3, only AR17 and GATA15 had differentially abundant proteins that had annotations involved directly in floral development. So we see from this independently that AR17 and GATA15 are emerging from our functional analysis as the 2 most likely candidates to actually be responsible for sex determination. So I'll highlight the results from those 2 in a bit more detail. So these are the results from our expression of AR17 in Arabidopsis. So here is a volcano plot showing the abundance of the proteins we detected. So we detected over 16,000 proteins. Of these, over 4,300 were differentially abundant. And among those differentially abundant proteins, we saw multiple genes that had annotations directly involved in floral development, many of which were annotated as being involved in pollen and tapetum development, tapetum being the precursor tissue for annotations. And notably, we see downregulation of many of these male floral development genes. We also see a downregulation of pistillata. So pistillata is a B-class matchbox gene. It is essential for development of male floral tissue. So if you remember from one of the earlier slides, I showed you that in persimmon, it is actually a downregulation of pistillata that is in the pathway of converting from male to female. And actually in poplar, where AR17 is the single gene master regulator, it's also thought to mediate the switch somehow through downregulated pistillata. And we see the same thing appears to be happening in shrub willow. So taken together, this tells us that with all these male genes being downregulated, that AR17 might have a role in suppressing male floral development. And this is consistent with what we see in our monoecious willows, where these 4 copies of AR17 are absent and we get male floral development. Now, GATA15 was the other gene that we saw differentially abundant proteins that were involved in floral development. So we once again detected around 16,000 differentially abundant proteins, about 60,000 proteins total, nearly 6,000 of which were differentially abundant. And this was the most of any of the genes we tested. And among those 6,000 were also multiple floral development genes. I'll particularly call your attention to these 2 isoforms of Lunig or rotunda 2 down here. So in Arabidopsis, when Lunig is knocked out, that actually results in conversion of sepals and petals to carpels, as well as a reduction in stamen development. So that tells us that maybe downregulation of this gene, which is what's happening, could be responsible for improved carpel development. Moreover, because GATA15 is a transcription factor, we were able to use DAP-seq to test its binding sites in the genome. So how DAP-seq works briefly is you express your transcription factor in vitro, and then you incubate that with genomic DNA. So that genomic DNA has the methylation profile from whatever tissue it came from. So in our case, we used female floral DNA and male floral DNA. So we have because we know that methylation, I showed on the earlier slide, there's a huge difference in methylation between males and females. So, so you incubate your transcription factor with your genomic DNA, you pull down DNA-bound transcription factor sites, and then you map those reads to your reference to get predicted transcription factor binding sites. So here, these are the consensus binding sequences from female on our left and male on the right. So you'll notice that there's very high support for these binding sites. They're pretty consistent between the two, between the female and male libraries, and they're also consistent with a GATA class of transcription factor, which should bind to a GATA or similar motif, which is what we see here, TGATC. So that tells us that our DAPSig method was valid, and we found 150 gene targets in the female library and only 28 in male. And among those gene targets in the female library were a couple floral development genes. So one is ATH1, which is a homolog of ATH1, which associates with A gamus, a class C Maslow gene involved in carpet development. And the other gene was a transcription factor involved in floral sensors. So together, this suggests to us that GATA15 might have a role in promoting female floral development. And that's also consistent with what we see in our monoecious willows, where GATA15 is the only candidate sex determination gene still present in those monoecious willows. So that leads us to our refined model for sex determination. So we took this integrative genomics and bioinformatics approach to identify candidate genes. We refined those models in monoecious willows, and then we did functional analysis to confirm the role of each of our candidate genes and arrived at this model. So notably, we think in Salix purpurea, sex is mediated by a two-gene system, such that involving AR17 and GATA15, such that AR17 is suppressing male floral development, while GATA15 is promoting female floral development. And so what that looks like in our model is males are at ZZ genotype. They don't have a sex determination region, so they result in male floral development. We think this is the sort of the default pathway for willow. When you have a ZW, you have a W chromosome. So that has a GATA15 that is promoting female floral development. It also has those 4 copies of AR17, which we think are suppressing male floral development based on our functional analysis results. So together, you get female catkins. Now in monoecious willows, which have that large deletion on the 15W, we still have GATA15 promoting female floral development, but we're missing those 4 copies of AR17. So we don't get the simultaneous suppression of male floral development. And as a result, we get male and female flowers developing on the same catkin. What's notable about this system is it is distinct from the single gene system in poplar, which is mediated by AR17, where that single gene alone can mediate the switch. So now shifting gears a little bit from the sex determination story, I want to talk a bit about a couple other projects I did in my research, also in conjunction with Oak Ridge National Laboratory this spring. So the first involves the assembly and annotation of 11 shrub willow genomes. Now, in the Cornell Shrub Willow breeding program, we utilize a variety of shrub willow species to develop cultivars. So willow, it's readily able to intercross between species. Now, up until this point, we were really relying on 2 reference genomes, which are actually shown here at the top of this table. So 9406, which is the Salix purpurea female, and Fish Creek, which is a Salix purpurea male, and is also the son of 9406. So we had a male and female reference we were really relying on. And most of the analysis I presented prior to this involved mapping to the 9406 female reference genome. But of course, we wanted to characterize, we wanted to sequence genomes from these other species, and so we could do some comparative genomics and add these to sort of as a resource for our program. So we selected 11 individuals across 6 species, so Salix purpurea, Salix suchoensis, Integro, Corynagi, Viminalis, and Udensis. And I actually have most of those displayed here, so you can see some of the diversity across the shrub willows that we're collecting here. So we did nanopore sequencing; those were then scaffolded with high C, and then Illumina sequencing was used to polish those assemblies. Illumina nanopore and RNA-seq were also collected. Illumina and nanopore RNA-seq were collected from each of these individuals as well, from 8 tissue types, which we use to annotate these reference genomes. So this table here shows some of the genome assembly statistics. And so I have the top 2 here. I have our prior 2 genomes as a comparison, and we see that our booster score is above 95% for all of these genomes, suggesting that they are pretty good assemblies. Now, if we look at the annotation, we can see that we're getting around usually between 30 and 31,000 genes annotated per genome. This is a little less than the about 34,000 we expect from our previous purpurea references. However, this is probably due to the fact that we did not collect floral RNA-seq or RNA-seq from stressed tissues in these individuals. So we missed out on a lot of the genes that are expressed in those very specific contexts. Nevertheless, these are pretty substantial genomes and annotations, and they really add a lot to the willow genomic resources. Notably, this is the first time anyone has assembled an Integra, Koyanagi, or Udensis genome, which we now have available. And I should also mention that a previous grad student in our lab, Justin Wilkerson, did a lot of work actually making crosses between each of these families and developing genetic linkage maps. So we have a lot of other genomic resources as well on these particular individuals, which sort of now form a nice core set that we can use for future studies. Now, in looking at these genomes, as you all know by now probably, I'm really interested in sex determination. So I wanted to see, what does the gene copy number of these candidate sex determination genes look like between different genomes? And so these are Vetrix shrub willow species. They all have been confirmed to have a chromosome 15ZW sex determination system. So we expected going into this that we would see the same gene content across the sex determination regions. We found out, however, that that does not appear to be the case. So I have AR17 and GATA15 are two strongest candidate genes highlighted here. Remember, AR17 is our predicted suppressor of male function, and GATA15 is our predicted promoter of female function. So from these, we see that both of these are present in Purpurea and Suchoensis females and absent from males. However, GATA15 is actually absent from our saved Integra female, and then AR17 is absent from the Viminalis and Koyanagi females, as is GATA15. So we see one or two of our candidate sex determination genes are not present in some of these other species, which was very puzzling to us. And that tells us that even though these have the same locus and the same chromosome involved in sex determination, that they probably have a different gene or set of genes regulating sex. And it's quite interesting and really sort of underscores how dynamic the sex chromosomes are within this genus. And so more work needs to be done in the future to confirm that and to validate what are the actual candidate master regulator genes in some of these other species like Salicaceae. Another interesting finding from the study was this Integra female P336. We found early in our breeding program that it appeared that all the progeny developed from this parent were female. So to test that, we crossed P336 with as many male reference genomes as we had. So we crossed it with 9401 in Fish Creek. These are our male Purpurea. We crossed it with 9403. That's the Monoecious Salix Purpurea. So the one with the weird disrupted sex chromosome. We crossed it with Suchoensis, with Koyanagi, and with Udensis. So 6 male parents across 4 different species. And in all cases, so we had over 200 progeny, most of them were flowering. And in all cases, we saw 100% female progeny, regardless of the male parent, which was quite fascinating, right? That's not normal to have an all female phenotype. So where it got really interesting when we looked at the next generation of progeny. So we took an F1 from this Integra by Suchoensis cross, and then we back crossed that back with Suchoensis to create a back cross 1, and also outcrossed it with 941 and 6 Purpurea to create a 3-way cross. And in both cases, once again, 100% of the progeny were female. This is after a second generation. So that was very interesting to us, right? And so our hypothesis of what might be mediated is that it could be a cytoplasmically inherited factor. So if you'll recall, chloroplasts and mitochondria have their own genomes. They're also only inherited maternally, so only the female parent can pass on its mitochondrial and chloroplast genome. So we predicted that if there were some factor in either the mitochondrial or chloroplast genome that were inherited to the female, that were resulting in all female progeny, that might explain this inheritance. So when we looked at our genome assemblies in the mitochondrial genomes, we noticed that this RPL10 homolog was missing from the mitochondria in P336. Now, RPL10 is a highly conserved gene. It is present in almost all plant taxa, including bryophytes and gymnosperms. So it's highly conserved, and we see it in our other willow genomes. So the fact that it's missing is quite interesting and definitely not expected. Moreover, RPL10 actually shows very high levels of expression in pollen in Arabidopsis and a couple other species. So taken together, this emerges as our most likely candidate right now for being responsible for this all-female inheritance across multiple generations. Now, we still need to functionally validate it, but yeah, this is a very unusual inheritance mechanism that we see, that we're able to elucidate with some of these whole genome sequences. So now I want to finish off the talk by talking about another project I did this spring at Oak Ridge, where we looked at developing protoplasts for transient overexpression and CRISPR knockout in shrub willow. So as I mentioned earlier, we cannot generate stable transformants in willow. Many people have tried, many people have failed. And so because of that, our functional genomics resources for shrub willow are quite limited. Indeed, many times you have to, the annotations in these genomes are actually based on their homology to poplar or Arabidopsis. And with our testing our candidate sex formation genes, we had to use Arabidopsis, the heterologous system, to test this. So I wanted to see if we could potentially use protoplasts as a means to do some in vivo functional studies in shrub willow. So we took 2-week-old cuttings. So willow is very easy to propagate from greenwood cuttings. We took young leaves that these arrows point to, took these leaf cuttings, and then used these to isolate leaf protoplasts, which are shown here. So we obtained very high yields from these young soil-grown leaves. And that alone is very interesting because in poplar and many other species, you have to use tissue culture grown leaves to obtain protoplasts. In willow, we can get very high yields just from these soil-grown leaves, which are incredibly easy to generate. So already willow is emerging as a very robust system for protoplast assays, more robust than even poplar. So we were able to express 4 different fluorescent reporter proteins. So in panel A, this is EGFP. Panel B, this is superfolding GFP. Panel C is GFP-UV, and it has this faint signal, which is actually what you'd expect from a GFP-UV construct. And then panel D is M-cherry. So all 4 of these fluorescent reporter proteins we were able to transiently overexpress. We obtained fairly high transfection rates and expression rates. So that shows us that transient expression is viable and works quite well in willow protoplasts. Also, this is the first time anyone has shown transient expression in willow protoplasts. So we showed we could isolate protoplasts effectively. We showed we could do transient overexpression. So our next step was to see if we could accomplish gene editing in shrub willow cells. So what we did is we selected 3 different single copy genes from the willow genome, Angustifolia, Phytochromae, and Frigida, and then designed 2 guide RNAs to target the first exon of each of these genes. So that's shown here in this cartoon. These red bars are the target sites of each of these guide RNAs. And so we did amplicon sequencing on those protoplasts, and those results are shown here. So this top line on each set is the wild type sequence, and we have each of the other sequences we detected and the number of amplicons of those. And then highlighted in red is the PAM sequence, approach spatial adjacent motif, where the guide RNA targets. So we can see either substitutions or 1 or several base pair deletions immediately adjacent to the PAM site for all of these, suggesting to us that these are in fact CRISPR edits that Cas9 and is working, and the guide RNAs are targeted in the willow genome. And notably for Angustifolia, our target sites were close enough to each other that our amplicons were able to span the distance between both of them, and we were able to detect multiplexing. So in this case, we see cuts at both guide RNA sites, resulting in a 90 to 100 base pair deletion from these genes. So that tells us that not only does CRISPR work in willow, but we can accomplish multiplexing. And so when we think about how now we have transient overexpression well established and CRISPR knockout well established, it really opens up a whole new realm of possibilities for functional genomics in willow. We can think about applications now where we might be able to knock out or overexpress a gene in willow protoplasts and then do single cell sequencing to look at in vivo expression differences, which we haven't been able to do in willow at all in the past. Or we can think about things like bimolecular fluorescence complementation, where we look at protein co-localization in willow cells. And these can now be done in willow cells with willow genes for the first time. So to summarize some of the results, so we did differential expression eQTL analysis to identify candidate sex determination genes. We used monoecious willows, crosses, and sequencing to refine our hypothesis. Then we did functional analysis on our candidate genes using expression in Arabidopsis and then DAPC. We conducted whole genome assembly and annotation of 11 shrub willow species, and found that the sex determination system likely differs between related species. And then lastly, we demonstrated that protoplasts are a viable system for functional genomic studies in Willow, and we now have this resource. So with that, I'd like to acknowledge all the people who helped with over the past 5 years. There are too many to put on the slide here, but all the members of the Smart Lab, some of whom are listed here, all the people I worked with at Oak Ridge National Laboratory this spring, they were instrumental in actually the last 3 of these chapters I presented. They had some role in that. So Jerry Tuskin, Jay Chen, Xiaohan Yang, and Bob Heddick in particular. And then of course my committee, all of them are here in this room. And then my funding sources over the years. So an NSF grant that Larry received that supported my research my first couple of years, the USD and IFA fellowship, which has supported my research these last 2 years, and then the DOE SCGSR fellowship, which provided the funding for me to actually go to Oak Ridge and receive a lot of the specialized training that substantially advanced my PhD progress. So with that, I'd like to open it up for any questions.
Thanks, Brennan. We're going to start with questions online. So if you're on Zoom, feel free to unmute and ask your question.
That's in willow. And so that actually 2 questions. What is the gene specific, tissue specific expression pattern of AR17 in willow? And the second one is the Arabidopsis ARR17 deletion line, is there any phenotype, like a flower organ differences?
Right. Okay. So I think there was 2 questions there. One is, what is the tissue-specific expression of AR17? And the other one was, what does the Arabidopsis AR17 knockout or overexpression look like? So for the tissue-specific expression in shrub willow, AR17 is highly expressed in female catkins. One thing I didn't mention is that there are also a couple autosomal copies of that gene that are present in males and females that show a low level of expression in both male and female catkins. However, we see much higher expression rate in females, presumably from those 4 copies in the SDR. There are low levels of AR17 expression in some of the other tissue types, but it's quite minimal compared to what we see in catkins. So I would say it's primarily catkin expressed. Now, as far as what does the Arabidopsis mutant phenotype look like? When I was looking at the existing literature, I could not find any. Well, they have generated AR17 knockouts. They did not really generate a substantial phenotype from what I gathered. So they're thought to be involved in cytokinin signaling, but they're certainly not critical, or they might have redundancy within the Arabidopsis genome.
Yeah, thank you.
Other questions online?
I have a question. I have 2 questions, actually. Brennan, first of all, good job on the presentation and the seminar, and an amazing amount of work. So congratulations on that. I had a question on the eQTL Circos diagram. What's going on chromosome 4 on the eQTL ring? It seems like there's a disproportionate change in, yes, right there at the bottom right.
So which chromosome?
Chromosome 4.
Right. Okay. Yeah. There's a huge number of eQTLs.
Yeah.
Yeah, we don't really know. Like I said, one could spend a better part of her career looking at all of the weird things that we see in this. So again, I was really focused on sex dimorphism and sex determination. So we need to go in and look at that more closely. I don't know what's going on there.
Yeah.
But you spend like half an hour looking at this, you can find all these weird little things like, oh, what's going on there?
Sure. It's always nice to have more work to do.
Right.
And then I had a philosophical question. With the consistency of Daiichi in the Salicaceae family, it's really surprising that there's so much differentiation and diversity in the mechanisms of sex determination. Chromosomes vary, ZW versus XY, et cetera. So give me a hypothesis or a philosophical explanation for why it's so tightly controlled in terms of Daiichi, but why the mechanisms vary so much.
Yeah, that's a good question, and a lot of people are, trying to answer, right? I don't think we don't know the answer. One sort of hypothesis is that, so there is obviously a fitness advantage to being dioecious in Salicaceae, right? That's why we see it. That could be because of traits that are sexually antagonistic. So where presence in one sex is detrimental, or presence in the other sex is beneficial. So we could think of things like secondary metabolites in catkins as an example, right? So these can increase pollinator attraction, but they can also increase pest attraction as well. So that's one example. Also, secondary metabolites take a lot of resources to produce, so they might not be beneficial to have in both sexes. As far as why has the STR shifted locations in heterogametic systems, the general model for how that could happen is if another locus somehow becomes more, there's a selection towards another locus because it confers some sort of advantage, right? So we can think about, if we think about like the system in Salix purpurea, where we think it's AR17 and GATA15, I would hypothesize that AR17 at some point translocated to chromosome 15, and by being linked with GATA15, that was advantageous in the ancestor of Salix purpurea, and so that took over as being the primary locus for sex determination, if that makes sense.
Very good, thank you.
Yeah, Jerry, you just stole Steve's main question, so.
Sorry about that.
Any other questions online? Okay, if you have a question, raise your hand and we'll come back to you, but I'm going to turn to the room here except for his committee members. Additional questions? It's your last chance with Brennan. Come on.
Yes, Brennan, can you tell us what the phenotype for the overexpression line for the two candidates? I think you tried more than 5, right? What are the phenotype for these different candidates?
So the question was, with the Arabidopsis expression lines, what were the phenotypes that we observed? So as you mentioned, we detected around 4 to 6, we obtained 4 to 6 events per gene. The reason we looked at proteomics was because even going into this, we did not expect to see floral differences in floral development, and indeed we didn't have the resources to go in and dissect all these tiny Arabidopsis flowers and look at the carbon counts. So from the start, we chose to look at proteomics because we didn't expect to see any noticeable differences, and indeed, in terms of floral morphology, we did not. The only kind of anecdotal phenotypic difference that I observed was the GATA15. That's our predicted female promoter gene. Its overexpression Arabidopsis lines did seem to have really poor growth. We had a really hard time even obtaining enough plants to test. That could just be a coincidence, or it could be a result of overexpressing the GATA15. We can't really conclude that, but that's the only of the 2 candidate sex determination genes. That's the only phenotype we saw. The other phenotype I'll mention is we overexpressed flower locus T as a control gene, and those Arabidopsis showed very early flowering phenotypes, which is consistent with Arabidopsis FT overexpression.
I'm surprised the whole genome annotation is only like 36,000 genes. You have 24,000 DEGs just on that slides. I mean, between female and male flowers.
So,
but that's a lot of DEGs, right?
oh, so you're saying you were surprised that there are 24,000 different
Yeah.
expressed genes. So I will add that is genes and alternative transcripts, but still it is a large number. But again, if you look at, let's go back to the catkin picture. If you look at female and male catkins, right, there's a lot of morphological differences between those. So it's not entirely surprising that we see widespread.
The common one is actually much less.
Sorry?
The long DEGs is much less than DEGs between these 2 different kinds of flowers.
Sorry.
The non-differentially expressed.
Yeah,
is
long
less.
differentially is
No,
much
the
less.
non-differential express is much lower. I think we detected around 36,000 total genes and alternative transcripts, and 24,000 of those, so about 2/3 are differentially expressed. Only about 1/3 are not single.
Yeah, but this whole genome is only like 36,000 annotated genes, and
34,000.
they are all expressed in the flowers.
So the whole genome is about 35,000 genes. It's about 57,000 genes in alternative transcripts. So we looked at the transcript level. So yeah, about 57,000 transcripts. 36,000 of those were expressed. 24,000 were differentially expressed. Now, I'll also add that our sample size was huge. We had 90 males and 90 females. So we were able to detect even small differences in expression for significant.
And since
It's a
popular,
very large data set.
and since you said willow and the popular are so similar, can you do the transformation of your candidate gene in popular?
So yeah, you bring up a good point. So the question was, can we do transformation of our candidate gene in poplar? The short answer is yes. Transformation systems have been well established in poplar. However, the challenge behind that is we want floral phenotypes, right? And if poplar is a tree, the generation time is several years. So one can imagine that's not very feasible in a PhD timeline. So in terms of resources and time frame, Arabidopsis is really the most viable option.
Okay, so I have one more question.
Go ahead.
Yeah, so I mean, auxin and cytokinin, they usually work antagonized. So I wonder if you test, what happened if you externally applied auxin to the flowers? What could happen?
Right. So you bring up a really good question. So one sort of pilot study I did a few years ago was I took floral shoots before the catkins emerged and actually applied, I think, auxin, cytokinin, gibberellic acid, and I think ethifon to them. I wanted to see if any of these hormone treatments would change the development, and it didn't work, and we didn't really see any differences. So we would have to test it at different developmental time points, right? But at least our preliminary study did not yield promising results. We chose not to pursue that further.
I would add that the challenging point there is when sex is determined in willow, and you have to apply that treatment at the time that sex is determined, and it's very difficult to understand exactly when sex is determined during bud formation, several months before the catkins actually emerge.
Right. So yeah. So floral buds are set in the summer. So willows flower in the spring. Floral buds are set the summer before. And I think in upstate New York, they are able to see, like, histologically the difference in male and female in around August. But that's when you can actually observe it. So it's obviously been initiated before that, and it's going to vary based on the factors.
Excellent question. Any more questions?
Yeah. So just from like a bioenergy, biomass perspective, do you see like differences between males and females out where they're growing?
Yeah. So in terms of qualitative differences, the only qualitative difference is the morphology. A previous grad student, Fred Boker, did do some work on the some quantitative traits. So things like crown diameter do appear to differ somewhat, and also I think rust susceptibility maybe a little also differ. Differ a little, like quantitative between males and females. It's not something you can go out and look at the field and be like, oh, that's that has a wide crown. It's a female. You can't. But so there are some quantitative differences related to biomass yield, but they are small.
So the good news is that Brennan will be staying here in Geneva. He's getting grilled by his future boss, Tyler, here. So he will be staying as a postdoc and switching to work on him. So we will look forward to continuing to see and work with Brennan. But for now, please join me in thanking Brennan for a great seminar. And this brings Defense-a-thon to a close.
This has been a production of Cornell University, on the web at cornell.edu.